should get another one in your email as well.
Okay, very good. Thank you. I already let Dr. Petroni know that we do have to leave a little bit early. So we're going to sit right by the door. Stephen and myself, yes. Were you at CIS this weekend?
I wasn't. I was in Boston.
Oh, that's
My
right.
granddaughter.
That's right.
How'd it go?
Really well, I think.
Yeah, no, I had told our scheduler when this baby is born, I'm going to just change my schedule, and so I just canceled 2 days of work and
Right.
left.
For sure.
Yeah. Got here just in time for breakfast.
Coffee. Is everything okay?
Yeah, let's open them up.
There's no dietary restrictions, is there? Among the... I know that.
Oh, wait, yeah, we had a discussion about that, didn't we?
I just knew that from, that there's certain...
Farmers Festival.
Right, yeah.
I just wrote that.
Yeah, I know. We just keep it, yeah. So what is her name? That's really pretty. Life.
Yeah, I didn't like it when I first heard it, to be honest. It's grown on me.
It's one of those, I feel like as soon as you see the baby and that's her name, it's just the way it is. And then it just works. We didn't tell anybody our name because we were like, I really don't want anybody's feedback. It's Brooklyn and if she gets here, that's the way it's going to be.
And are you aware of the suggestion that Tillis is supposed to present on a day when he's out of the country doing something with the college?
Mm-hmm.
So he was supposed to talk basically about an update on food, research, food. So I got an email from Mike Weiss the other day suggesting that you take that spot instead. And he said Lisa, but I don't know. Did he mean Lisa Winter? What does she have to do with the study?
She wants to get involved, so she wants to step up, to get more involved in doing everything.
So that would mean to me that the date that Steve is scheduled, I think, is April 25th. You would be presenting again with her. Mostly anything you want, as long as it's a good presentation, talking about what we're doing over now, what you're doing with developing oral desensitization protocols. Probably use it for advertising as well as to what studies are currently ongoing. But you know,
We have plenty of stuff going on.
yeah.
Make sure
But I think you got to go to the literature a little bit too, and maybe pull up some stuff from the meeting.
Okay.
I heard Gideon Lack talk at the meeting. He didn't really say anything new, but
There is nothing really new out there right now. There's a lot of talk about stuff, but
I guess you could.
Good morning.
Good morning.
Is our speaker today?
Yep. I guess you could quickly re-review the change in recommendations about peanut.
what's been surprising is how difficult it's been to implement to the
Right.
community.
Great.
Pediatricians have been very resistant to any sort of change. It's been very hard. The message has been trying to get out there, but
Well, anyway, I'll plan on two of you doing that for Steve.
I can do.
Morning, man.
How's things?
Not bad.
I'll
I
show
got
you
a
a
sore
picture of our
knee.
granddaughter.
Sure. No, I'd like to see it.
Just back in Boston.
Oh, you went
visiting.
back there too. Good for you.
Just last weekend. Just came back Sunday, Saturday.
Interesting way to print it.
I have to get the holder.
Yeah, the last picture of me holding. It's Johnny and the dad.
Looks like he loves it. Okay, it's something I get attached to the other side of the critter. It was worth the wait, huh?
Yeah. So we didn't know if it was going to happen, as you know.
Yeah, that's great.
That was good.
I'm miffed. I had a sore knee. I haven't been able to run for a week. You know what I think I did? Being the amateur orthopedist, I think it was a little, like Baker's cyst that ruptured. Because it's in the back of a weird place. Doesn't feel like in the joint.
That's where you had those, right?
I know. I couldn't feel it, though. It's not like it's big enough. But I remember for about a week before this, it just felt a little funny back there. So I was running, and it just felt a little weaker, not painful. And then all of a sudden I'm running just like that. And it hurt like crazy. I had to walk. So it's getting better. Most of the time, I understand, if you pop them, takes care of it. It's still a little sore.
So does it hurt to walk?
No. Actually, I'm fine walking. Going downstairs still hurts. I tried running yesterday. I couldn't do it, but it's better. I hope, you're waiting for that day that you can't do it anymore, just say that.
Well, you know what it is. It's getting old. Like it or not.
I know. And I'm hoping to rebound from this. But
I know. I heard when I get up every
that might
morning.
be true, too. What's that? You're reminded every morning.
Every morning when you wake up, I heard here. I heard there. It takes a couple hours before
the kinks out.
the kinks come out. Got any trips coming up?
No, but I'm looking for South American or Antarctica. I'll let you know.
Well, if we go to
Hey, I
South
got good
America-
news for you.
What?
I'm doing this research on space travel and everything.
Yeah.
Usually, as long as you can put in about 3 or 4 days, you get over your motion sickness. You have bad motion sickness.
You want to put me in space, in other words?
On a ship. But you just have 3 or 4 days of misery, and then you have fun.
That's the life. Those days of misery you don't forget.
No, I never-
It's a uniquely unpleasant experience.
No, I'll never forget that drive up to Big Sur.
You won't forget it. Imagine how I feel. How are you, young man?
Yeah, we were driving up Big Sur. Decided to go up the hillside, which is very steep. The road's like this. He was driving even, and all of a sudden he turned green. And you couldn't stop at that time. They had fires in here. So you can't stop. There's a state park or something like that. Anyhow, we get to the top, kind of drag him out of the car, lay him on the ground. And the forest ranger or whatever comes by and says, you're not allowed to stop here because there's fires in the area. And I said, yeah, but look, he's got motion sick. He said, You can stay. It's all green you look. I don't know. A whole bunch of those Dicker pine cones. Isn't that beautiful? I know. They were huge. I want to go back and get some more and bring them home, though.
Well, next time we go down to Carmel, I'll give you dates ahead of time so we can get down together. Spot some more birds.
No, absolutely. I'll tell you the other trip. But again, it's on a ship. The Northwest Passage, now with global warming, you can go through. That sounds like a lot of fun. Go, you start in
Oh, I started to say, if you go to South America, you got to go to the falls that we went to.
Iguazu. Yeah.
Well, that's amazing.
I've seen a picture of it. It looks incredible. You know, you have to do that as an add-on trip. Everything goes from Santiago to Buenos Aires, and then you have to add that.
Yeah, you can, it's a short flight. I forget where you fly into, but you're almost at the juncture of Paraguay, Brazil, and Argentina.
I have two
all
pairs.
three countries.
Oh, I like that. No. I got my travel agent working on both those trips. The only thing is the one Northwest Passage is like 15 grand per person. And that's the cheap seats too. You know, just so expensive. But it goes all the way from almost to northern Alaska, all the way to Greenland and all those areas. So you're going like 1,500 the old days. So you're covering a lot of
Where does it start?
You know, no, it's not as far. It starts in one of those villages for the Eskimos way up there. It goes and finds the ruins of the Erebus that they only found about 3 or 4 years ago. You know, they knew that people had gone up there trying to find the Northwest Passage, disappeared, and never came back. About 3 or 4 years ago, they found them, their ship, and the remains of everything. Obviously, they all died there. And so part of the
What was the name of it?
Erebus, E-R-E-B.
Was that the name of an expedition?
Yeah. But yeah, well, it might have been the ship. I've been the ship for this long. I'm not sure. Either one. But anyhow, you stop there and you can see all the ruins. And the big thing was they just found it like 3 or 4 years ago. So. Traffic's not bad headed that way.
Yeah, wasn't that guy who came last week just trying to make it?
Get out of this area. Everything is autoimmune, I think, now. Even asthma is autoimmune. Yeah, no, it's interesting. And Nereus might be. Of course, I tell all patients hives they have autoimmune. Pretty much. That way they won't go searching and say, you don't know what it is? Well, we're not certain. It's idiopathic. I always say, you know why we call it idiopathic? Because your doctor's an idiot and he can't figure out what it is, you know. But no, I said, this is autoimmune. You don't have to keep searching, go for doctor.
We had a young woman, just 15-year-old.
Yeah.
I guess it's good that I'm getting these cards in months, but no, but I have no choice. They're taking mine. This is the diesel.
Usually autoimmune. Very strong flu. And someone else, which generation, female.
You sell it back. You have to get it.
Went to the mother. Just see why it didn't. I'm guessing you're back.
I thought that was
But you can try to do something.
Get your credentials.
no pressure.
Yeah, I mean, I look like a star. Just, we all know why. It was easy to look into.
You look like a star.
You know this guy? Not a star.
Yes, but I came over to read Lexi to sleep on the Lexi's 3.
It's based on a mix of
Smart.
year, mileage.
You know, shouldn't compare grandkids.
No, it's because Sarah works.
Frankly, it's actually
She works with her. Yeah, no, she picks up early, but she's learning to play the piano. She's going to have culture that none of us have.
Aileen was reading when I was at the show. Her dad, so Gayleen was reading a Dr. Seuss book that she had read
essentially it's
to
an
Jonathan, when he was a baby.
$4,000 divided by 9 years.
I mean, my parents, they had a 17-year-old
Why you don't grandpa?
and
Or is that the picture?
Yeah.
Look at Jonathan too.
And like drive them to a dealership without a Civic. So they got $5,500 for the van.
It's interesting. She's such a small baby, John.
Civic. So for $12,000 or something.
Yeah. It's like fantastic.
Still using.
Fortunately, the kid's got
$30,000.
mother's numbers, not Jonathan's numbers.
So just.
It was a great deal. People who cashed in on
Yeah, a lot of
it.
stuff babies aren't.
Yeah, well, I was.
You know, you always say they're cute, but they really
Oh, you
are.
remember
It's
the
Seinfeld,
The Seinfeld. You gotta see the baby.
breathtaking.
How long are we out there? Come see me quite.
We left on Thursday. I just, told my clinic I'm not working. I got cancer. So Thursday, Friday, Saturday. We always have trouble getting back from Boston. It's late. I was in seat 6C because we fly a lot. And they send me the day before an upgrade. And they cancel it. They substitute a smaller plane that doesn't have any first class seat. So I get to the airport. Well, you're not first class anymore, and you don't have your sixth row seat, not even row 17, because we gave away your row 6 when we moved you to first class, which we don't have anymore. And this is a great deal. I'm gold with you. I fly 40, 50,000 miles. They actually put me back in first seat. Oh, yeah. We both went, and
Remember the days you used to fly separately all those years?
Yeah, that's nice.
It's hard enough to make a link connection.
I remember
Things are over though now.
I meet with pharmacy again on Thursday, so I'll be there.
We only get
Rather like taking.
And I couldn't get back in.
Yeah,
I think so.
Let's go ahead and get started. Everybody take a seat. Word of the day is repair. Dan's going to present gene therapy and repair, but there's two comments. One of them, I'm thinking about the year-end session. I think it's last Tuesday in May. And I think we're going to have it in the adjacent building next door and cater it over there. It's a little cheaper and more efficient than doing it up on Upper Campus as we've done it the last couple of years, but I'll give you particulars. The other thing I'm just going to comment as I was driving in here this morning, I was listening to NPR. I don't know who else was listening to NPR. This is not political. You're not supposed to say anything political in these sessions. They were talking about lying. A neuroscience study that the more you lie, the more your brain ignores it. I don't know if that means anybody in... Anyway, so let's go ahead. I've been called before for making political remarks. I've gotten in trouble. So shut my mouth.
All right, thanks, Lynn. So, Lynn asked me to give this talk about gene therapy and repair. If you remember last year, David gave a similar talk, which I didn't understand. So, I wanted to take it upon myself to learn this topic and what. So, one of the things I hope that you get out of by the end is what CRISPR-Cas9 and what can it do for us in the future, among many other things. So, here's my disclosure. I'm a sub-investigator, primary investigator on many different studies. There will be one study that I am that we are involved with that I will talk about. Apart from that, there's nothing else that I am involved with that will have anything to have impact on this. So the goals of today is describe gene therapy and repair. We're going to describe the various mechanisms of gene repair and describe how gene therapy can have some impact on us in the allergy immunology field. So what is gene therapy? Is gene therapy this? Not quite. So gene therapy is an experimental technique. It's been going on since the late 80s that uses genes to treat and prevent diseases. This can be replacing a mutant gene, this can be inactivating or knocking out a mutated gene, or introducing a new gene in the body to help fight a disease. Or it can also be modifying a cell that can be introduced to fight the disease. And we'll talk about that as well. So the simplest method and the crudest method to introduce DNA into a cell is by injecting simply naked DNA. This can be done into the muscle or into the skin. Typically, more modern techniques have been using plasmid DNA, with the idea that the plasmid DNA will then be integrated into the antigen-presenting cells and present the gene that you're trying to insert to be then presented to immune cells. There are several clinical trials using circular DNA. The idea behind it is this, is that the circular DNA is the same type of DNA that's found in bacteria. And what you get out of it is a Th1 response because it's circular. And so this idea is being used to treat allergic disease. So a company that we're involved with, that we're doing the clinical trial for, has introduced some special types of plasmid DNA with integrated allergy genes. Their first foray was with Japanese cedar, and they did this in Hawaii with Japanese Americans. Japanese cedar is a very significant pollen in Japan. Like 25-30% of the population suffers from allergic rhinitis from Japanese cedar. And it's a Japanese company, so that's what they did a phase 1 and 1A, 1B, and they found it to be safe. Of note, the patients who were on the actual treatment, their skin test disappeared. That was after like 2 months being on it. That's unpublished data that was from the company. The one we're involved with here in Seattle is the peanut study. Actually, I'm going to be doing a challenge at 8 o'clock today, so I have to kind of piece out of here not too quickly at the end of this talk to do one of the challenges to enroll into the study. And inside the plasmid DNA is the ARA-H2 gene, so the peanut gene. And it has passed phase 1A, which is just introducing the plasmid into healthy individuals. We're actually introducing this into confirmed peanut allergic patients, so all challenge-confirmed patients. We gave, here in Seattle, the second human being with a peanut allergy ever this thing. It was safe. And we're going to be doing to the second human being very shortly. For this initial foray is going to be all intradermal, and then the next step will be intramuscular. Viral vectors.
Dan, on that
yeah,
point, how long can you track that for how long, certain cells?
How long? Right now we're just seeing if they have, if it's safe. So right now it's just if they have antibodies to the plasmid itself, and then if it's safe or not. Whether it has any efficaciousness or persistence in the body, we don't know. It's definitely not permanent.
But in animal models, how do you track it to know kind of the long-term effect?
So they have published data on the Japanese cedar, which showed that it did express for a certain amount of time in mice. And it did have a demonstrable effect. They had some desensitization with the humanized, sensitized models for it. But still, this is really early days, so we have no idea what this is going to do. We're going to be doing our first post-challenge on this gentleman probably in a month or two. So we'll see what happens. I don't know what he's on, but I'm curious.
So how does it produce a protective immune response? What are you stimulating by inserting the gene?
So the idea is that the plasmid DNA will then integrate into the APCs, which will then start to express the ARA-H2 gene. It will then get chopped up and then presented via MHC2, and then presented to and simulate a type Th1 type of response to it. So overdrives it from the Th2. That's the theory. I don't know if this will actually turn out to be anything, but just like anything, you got to try. So it'll be interesting. Viral vectors are the next and most common way to introduce genes into the body. This has been going on since late 80s. Various viral vectors over the years have been used with two major approaches to it. One is in vivo, where they actually infuse it into the bloodstream or directly into the organ system that they want to have changes, or ex vivo, where they remove the cells from the body and then introduce the viral vector and then do the transfection and then return those cells back to the body. So this has been quite useful for us in terms of primary immunodeficiency and gene therapy. This is really great for monogenic diseases such as PID. So XSKID, RAGSKID are both diseases that are universally fatal without treatment. And right now, currently, stem cell transplant is the treatment for a significant portion of this population. Especially with newborn screening, we're really good at it. So the latest data I saw for safety in the U.S. And in terms of survival with bone marrow transplant, it's around 98, 99%. So we're really good. But appropriate matches, that's with an appropriate match. So, for instance, I had a patient who had CGD, and he was African American, but his father was from some unusual country that was an island off the coast of Africa. We did a global match, we found 2, and they were both terrible. So he was absolutely not a candidate for transplantation. What he could be a candidate, which would be for gene therapy. And so the very first gene therapy was conducted in two little girls with ADA SCID, both of which are surviving and are doing quite well. So the first kind of most common virus that has been used is our Gamma-Retroviruses. These have, if you remember if you're in basic biology, have RNA that integrates themselves into the host DNA. They are only able to integrate into dividing cells. This will be important later on. Its integration into the genome is random. And because of this, it may lead into inducing oncogene transactivation and therefore leukemia. So in the late 90s, early 2000s, there was an XSKID trial that used retrovirus that repaired CD34 cells. And then 4 out of the 11 kids that were in that trial got T-cell ALL. 1 died, 3 got better with treatment. But that kind of derailed this for about 15, 16 years. This has finally kind of come back. A lot of this stuff, there's been kind of slow progress because of bad stuff that's happened along the way. And that's what happened with this one, Adenovirus. This is a DNA virus, so it doesn't integrate into the host genome. So the problem with integrating into the DNA and causing cancer is not here. But it has very high immunogenicity. If anybody's ever been affected by adenovirus, you know how nasty a virus this is. And in 1999, a patient that was being treated in a gene therapy trial with OTC deficiency died. They looked back at what had happened during this trial. The primary investigator had significant financial involvement with the company that did this. He also did not release in the informed consent that there was many different animal trials where the animals actually had the same problem. And so didn't disclose that. And 2 other patients also had severe immunogenic reactions to it, also not disclosed. And because of this, adenovirus as a vector is really not being used in the United States anymore. It's really killed gene therapy in the U.S. for many, many years, up until very recently, as it kind of started to come back. And just with the additional safety guidelines and safety review stuff, we had to do the similar type of thing with just the naked DNA. Believe me, I never knew regulation of this extent ever. We've been involved with many different trials, but the gene trials and review of any sort of anything involving gene therapy, excruciating. They really review. So you have multiple levels of screening to be able to get through this.
What's OTC?
Ornithine transcarbamylase deficiency. It's a inborn error of metabolism. They have mega livers. They die early. It was 18 when he died. The latest generation is lentiviruses. Lentiviruses are things like HIV, SIV, and FIV. They can both express in genes that are both dividing and non-dividing. And a lot of work has been done in this. And so they have a much safer profile. They have their promoter sequences and their integrases have been modified so they selectively not integrate into those kind of more oncogenic areas. So it becomes less penetrance, less problems. And because of this, they become safe enough that we're actually going to be starting here in Seattle a gene therapy using a lentivirus vector. Andy Schneiderberg will be doing, who's the primary investigator here in Seattle, and they're recruiting for a SCID. And so they're using a lentiviral vector. So we'll see. They haven't got the first patient yet. But like I said, it's gonna. I was talking to him about this. It's going to be a hard one to recruit for because it's so good. The transplant is so good, and this is experimental. So it would be a very select group of individuals, of children who would go into this. Really, what I think this would be good for is the second phase of this trial, which will be in the failed transplants or the older kids who has kind of like a chimerism there, which makes transplant extra difficult. And this would be helpful for those folks. Bringing back to allergy. So this is a gene therapy. It's mice, but I thought it was an interesting little trial using basically creating an omalizumab mouse to treat peanut allergy. So they took immunodeficient skin mice and they were injected with serum from peanut allergic donors, all of which had a history of peanut anaphylaxis. So the mice were injected with an adenoviral gene vector that encodes for the high-affinity anti-IgE molecule, very similar to omalizumab. The animals were then exposed to peanut and to induce anaphylaxis. So this is the basic setup of it. They have the mouse, and they either transfected with the one encoding the IgE, anti-IgE, or the one that's encoding something that's null. Expose the sensitized skin mouse to the peanut and to see whether it has any change on anaphylaxis or not. So here we have three. There we go. We have three different mice here. So this is the control mice. All right. This is the anaphylaxis score in the x-axis. This is 2 weeks after the gene therapy, and this is the anaphylaxis score of the gene therapy mouse with the high-affinity anti-IgE, and this is mice injected with omalizumab. This is 5 weeks after therapy, and we can see that the persistence of the anti-IgE and protection from anaphylaxis is still there. And then the mice that were injected with omalizumab has worn off, and they have anaphylaxis. This is looking in a similar way, except for a level of histamine release in the body.
This gene is making total anti-IgE or peanut specific?
Total. So it's just a high-affinity anti-IgE molecule. This is a survival graph after exposure. So the mice died, but let's see here. So this is the yellow one is the control mice, and you can see that quite a few of them did not survive the anaphylaxis. The omalizumab mice, and then the ones with the anti-IgE built in. So maybe an interesting thought, but that's something. It's a thought process. And adenovirus is a temporary thing. So it's a DNA gene, so it could possibly be extrapolated to human beings at some point. Problems with viral vectors, while they are certainly getting better, they still will randomly insert genes into various parts of the genome. Cancer will always be a risk, though minimized. But what you kind of want, though, is if you're going to do gene therapy, is that you want to have the promoter and the inherent control of that gene to be in place. If you just randomly place it, you don't have that control. So really what you would really like is something like gene repair. So this targeted ability to insert a normal gene in place of a gene that is defective. And that's what this newer idea of gene repair and gene editing, much more modern approach to this. So, as I mentioned, it's a specific targeting of a specific sequence of DNA and cut at a very specific region. And these gene editors induce a double-stranded repair and uses our body's natural repair mechanism of the cell to induce either gene knockouts or insertion of a gene in the specific site. And this is the basics of it. So, essentially, you have a region that's specific for a DNA binding region, one side and the other side, with a nuclease attached to it that will chop right where you want it. Okay. Depending on the technique, the more specific the DNA binding region is. But that's the basics of this. And so you can knock this gene out if you clip it, and then the gene repairs it with a non-homologous gene repair. Non-homologous gene repair is much more error prone, and so if it induces a transcription error point mutation, frameshift mutation, then you will knock out that defective gene. It can also insert a good copy and replace a defective copy if you have a transcript that you kind of add into it. So those are the two basic ways that you can do this. There's currently 3 major techniques that are involved with this. Zinc fingers, TALENS, and CRISPR-Cas9. I'll talk about the first 2 in brief, but I'll talk in detail what CRISPR-Cas9, because that looks like what the future direction will be. Zinc fingers are DNA binding motifs that are attached to nucleases. So that's DNA-specific binding region that uses zinc fingers, which then recognize a certain part of the double-stranded DNA. They have some specificity, but not nearly as good as the later generation of things. One of the biggest limitations is that they're very large, and so this has to get into the cell itself, which makes it difficult. So it has been used in human beings. So the first gene editing trial was done in humans and utilized zinc finger nucleases on patients infected with HIV. We know that CCR5 is a very important co-stimulating molecule required for HIV infection, and then defects in this offers a protection against HIV. Certain Northern Europeans naturally have this, and so they have a resistance to certain types of HIV. So 12 patients were enrolled in this open-label trial with zinc finger modified CCR CD4 T helper cells, and these modified CD4 cells were then infused into the body. And what they were basically looking to see is that if these modified CD4 cells would have better survivability. This busy little slide here basically shows a couple of things here. Let me get my pointer out. The green one is the viral load of the HIV virus. The black is the unmodified CD4 cells, and the blue is the CD4 cells that have been modified with the zinc fingers. So essentially what they did is they infused these cells, and then they stopped these patients' antiviral drugs to induce an increase in viral load. What they wanted to see is how well these modified cells would survive relative to the unmodified cells. And as you can see, the CD4 cells that were unmodified, once the viral load went up, quickly died off. Okay, compared to the modified CD4 cells, which kind of stuck around and really did not significantly change it dropped by like maybe 1% as opposed to somewhere like 20% drop from it. Then they resumed the antiviral therapy, and everything kind of normalized. But it was just a basic proof of concept, just to show that, just to see this. Also, these are not stem cells, so they have a natural, they will go away eventually. Some patients became, 2 patients had basically non-detectable viral loads, even though they had very high viral loads before they started the trial. But it was just a kind of a proof of concept. They're not going to do this again. They're going to move on to other techniques, and so. And I don't think anybody's using zinc fingers anymore. So they're moving on to things like this, TALENS. TALENS are, if you look at the name, so TAL, transcription activator-like effector nucleases. So the TAL part is the one that recognizes the DNA, and the nuclease portion of it. Originally found in proteobacteria, and it was used to attack the genes of the plant to increase its infection rate. And so TAL can be customized to recognize a highly specific section of DNA, and with the nucleus break at a very precise location, more so than the zinc finger. This is the basic setup for it. The TALENS are being used to create, right now, chimeric antigen receptor, or CAR T cells. You've probably heard of CAR T cells. And companies are using this for T cell products. And these CAR T cells use these TALENS to knock out the T cell receptor and the CD52 receptor, as well as to specifically target the CD19 leukemia. The reason they did this is that CAMPath is very commonly used, and if you knock out CD52, you will prevent these cells from dying from CAMPath. So you can continue the chemotherapy that we're using on these kids, but use this cell to go in and hopefully home in and target the B cell ALL. In the UK, they were doing just basic safety trials for this. But 2 kids who were close to death, they did a compassionate use for these two children in 2015 and 2016. And so they did use these cells in them, and they both went into full remission. And now one is 1 year post, one is like 6 month post, and they're both still doing great. So proof of concept, interesting, still being investigated.
What do you mean off the shelf product?
So this is not modification of from their blood to make these. So this is T cells that are just being made, all right, and that they created and that they modified. So this is not their own T cells. So it's not modified from them. And so these T cells will have because it's not them, will be destroyed once their immune system is reconstituted. So it's not a permanent part of their immune system.
These are T cells that targeted CD19 B cells that
Correct.
killed them?
Yes. Yeah, so it's for B cell ALL. So, and CD19 is a prominent CD marker for B cells. Okay, so CRISPR, Cas9. So this is the kind of the latest comer and probably one of the more interesting versions of this. CRISPR stands for Clustered Regular Interspaced Short Palindromic Repeats. I'll show you what this means in a moment. It took me a little bit to figure this one out. Cas is the scissor that causes the double-stranded break. And this was originally a primitive immune system in bacteria and is specifically against bacteriophages and bacteriovirus. And has been modified by us as a system for gene editing. So this is the original CRISPR system from bacteria. So this is integrated into the genome of the bacteria itself, of the plasmid DNA. And so the clustered regular interspaced short palindromic repeats. So in the CRISPR gene, there's these palindromic repeats, so those segments there that are the same one way and the same backwards, and that they happen over and over again. Interspersed these were these genes, and for a long time, nobody had any idea what these were. What these are the history of this bacteria being exposed to viruses. So these are sections of viral bacteriophages that the bacteria has been exposed to and recognizes. And so it has the cutter there. And so these are the gene sequence and assortment. And this is how it basically works, is that the bacteriophage injects its DNA into the bacteria. And so this is the Cas and the CRISPR of the bacterial genome, and they secrete the Cas with the specific target. And so this will then clip onto the viral DNA and then cut the viral DNA right there to induce mutagenesis, so it will inactivate this viral DNA. So it's their crude version of the immune system. And if this was a brand new one, they'll just release a Cas to go up there, chop it up, then it'll integrate that piece of that DNA back into the system, and then it will learn that this is a bacteriophage, and then it will then integrate into this. So it's not just these three, it happens over and over and over again. So the more viruses this bacteria gets exposed to, the more this gets recognized. And this is Nobel Prize winning stuff. And so somebody thought, well, if it can recognize viral DNA, why not it can recognize a section of human DNA? So that's where the CRISPR-Cas9 system basically was created. It's from Streptococcus, and so the Cas9 is this big guy right here with the two nucleases, the clippers, and it has this modified section of what we call guide RNA. And this is what you can customize. And this is the section right here that is recognized in the DNA. Okay? And so this guy goes in and has a helicase and a nuclease to unravel and then be able to specifically target that section of DNA, and with the Cas9 to cut that specific segment of DNA. All right? And it can induce a double-stranded break. Now, if you stop there and don't have any guide DNA, then you can create knockouts. So a good friend of mine, he's involved with this, basically this NIH competition. I call it that, but that's basically what they're doing, is that the first lab to generate as many knockouts mice as they can will get this huge grant from the NIH. So they're in this giant rush. And so this is what they'll do, is that they'll introduce this, and they'll have many different segments, and they'll just chop and induce a knockout because of the mutation of that specific gene that they're targeting, right? If you want to insert a specific gene segment, you do the same thing, but then you have this host RNA that will then be inserted at that point, and then the gene will then be created right at that segment. Okay? So that's the basic setup of the CRISPR-Cas9 system. Does anybody have any
So the
questions?
gene uses the RNA as a template
Yes.
to make DNA?
Essentially, yes. And that will then integrate into this. Anybody have any questions about that? Is that wonderfully clear?
Easy.
All right, so right now, the CRISPR-Cas9 system is being utilized in humans in China, not here. So it's being conducted in phase 1 clinical trials. And they are, and there's many of them. If you go to clinicaltrials.gov, you will see numerous trials for advanced cancer, none of which is here, all of which is all in China. So people with advanced small cell lung cancer, different types of lymphomas, leukemias, bladder cancer, prostate cancer, everything. And what they're using are modified T cells with specifically PD-1 knockout T cells, which essentially will attack these cells. It will attack any cell, really. It kind of, you lose the control mechanism for these T cells to not attack self. So they are doing phase 1 trials. Now, we have approved the very first trial with this similar type of method here in the United States. It happened late last year, and it's with Sean Parker, the guy from Napster. He had this big gene therapy. He donated like $250 million to do gene therapy, and so it's using money from that to do this trial. The guy who did the zinc finger with the HIV, he's doing these modified T cells. Okay.
So what's the point of, go back a slide.
Yeah.
So here you have a very precise technique to take out a gene and insert the gene, and yet the first trial seems like very non-specific. You're just inserting a non-specific to activate killer T cells against anything.
Yes.
Why not use the technique in a more precise way?
Definitely. That's one of the limitations is this next part I'm going to talk about. So one of the limitations for this is one thing to edit at a small level, like a number of T cells that you're going to fuse at the million, billion level. It's another thing to modify an entire bone marrow or an entire organ system. These things have a hard time in getting into these cells, so transfection is a problem. So this massive scale, the scalability of gene editing is still yet to be solved. Some are using or think of using packaging these into things like the Lentivirus as the way to get into the system, then inject, and then you can have your specific target. But that has not been able to be extrapolated into humans yet. But they're starting to do that in other things. It's being utilized in agriculture, though. So people are using CRISPR-Cas9 in agriculture for gene modification. The other thing is relative fitness of cells is the other major issue. If the repaired cells are more likely to survive than the defective cells, then you have a permanent cure. So, for instance, in egg skin, so they don't produce T cells and B cells. So the modified cells are going to be relatively more fit than the unmodified cells. But in patients that have, say, like CGD, the neutrophils, whether they can kill bacteria or not, it doesn't matter to the body. So that they don't have a permanent graft oftentimes. So they've seen this in the clinical trials with XCGD, where they've repaired the cells, but they just are not a permanent transformation, and they just kind of slowly go away, and they have to redo this over and over again in order to do this. So there's work on using promoters and other things to kind of increase the efficiency of this, but that's still work to be done.
Dan, do they ever take this and just do like stem cells and take them out and use CRISPR on them and then?
That's the other kind of ethical thing too. So you can certainly do that on seeds and in basic animals, but then you have problems where, okay, so we can do this and we can specifically target and we can modify cells. should we do this at the embryo level? All right. So if you want to cure something and cure a genetic disease, and you can't do it on like 1 billion cells, how about we do it on 5 cells? And so editing and targeting gene at an embryo level is certainly possible, and we can do it. But whether or not we should do it is the other major question. Are we going to have tiered systems where we have, because it's not cheap. This is only the rich are going to be able to afford this. And so will only the rich kids be the genetically perfect? And then do we stop at diseases? Why not have, like, I would like to be 20/20 vision too. Or why stop at 2020, 2010, 25? I like that too. And then, so you could do lots of things. Whether we can do it or should do it, that's a very different question. And so the ethics of this are quite interesting. That where we can ultimately create designer babies. And then who knows what people will do. People are crazy. So maybe you want to introduce like tiger stripes to your kid. Who knows what people will do. Because why limit yourself to human genes? Why not the other genes? Okay?
There was some research the Chinese were doing with dead fetuses with genetic defects. They were trying to correct the defect and then see if they could raise the embryo up without that. Has that gone any further?
Definitely not a clinical trial. So definitely not something that I'm sure that people are doing this, and if we not to get on a political soapbox, but we cut the NIH budget by $6 billion, like some people have proposed, then we are going to be behind the 8 ball, and we're not going to be great. So genetic engineering can be sponsored in certain ways. So maybe Coca-Cola sponsor a baby.
It seems to be something like sickle cell disease would be a great target.
Definitely. So there's so many diseases. I kind of limited it to our stuff in AI, but for sure, sickle cell, different types of metabolic disorders. Right. Those are all kind of unilateral gene stuff. So there are many things that are certainly can be used for this trial, for these type of techniques. So it is quite exciting. And so gene therapy is coming. Whether it's going to be by us or by the Chinese, it will be coming. And will have significant impact to medical science and what ultimately will happen. In the not too distant future, that if we have more gene therapy, you will have kids that will come in with like a severe, that had SCID, who are completely normal. And so you'll get a lab report. Like, this is the number of modified cells. These are the number of regular cells that they have in their body, and that's their chimerism, and that's how you monitor your immunodeficient patients. And so it's going to be hopefully simple at some point. But yeah, that's it. Like I said, I have a trial. I have to do a challenge in like 20 minutes and avail it to you. I actually have to get my badge too, so I haven't been used since I was a fellow. So I have to get a badge so I can get in the building. Does anybody have any questions? Sorry, let's finish early. Comments or anything?
Yeah, there's a company called Juno that's about a mile from here that's doing a lot of the car stuff.
It's across, dude.
I mean, yeah.
Yeah, right across the street.
Most of the car stuff we do here, though, is using viral vectors.
Right.
Not using TALENS or...
Right.
I don't think anybody's, like I could see, most of them are in Europe, UK, or in Europe, or in China that are doing this. These are companies. So this is all company-based things. These are not universities. These are people trying to market this stuff. Yeah. So these are customized, humanized cells.
Is it not, though, going on at the NIH or being done in academia? Already at a commercial level?
Like I said, one of the issues with genes, anything gene stuff, is that the regulatory is amazing and significant. Mostly because of the historical deaths and leukemias that have been generated from it. It's really curtailed our own kind of gene research. It was really up until last year that the NIH was really, anything involving a gene, they would have a regulatory gene committee called RAC review, and it was excruciating. RAC reviews are pains. And they're finally kind of backing off on things that have been proven safe and other things where they don't need super high RAC reviews at multiple levels. You have to have RACs locally with your own IRB, and then at the central at NIH too.
So this
I-
is in
I-
China, that's obviously much easier.
Oh, no doubt.
Or,
Yeah, they're doing adenoviral type vectors. They're doing all the stuff that we've kind of curtailed. They're doing that actively and commercially. So there's commercially adenoviral vectors for humans over there.
All right, well, thanks, Tom.
Pause for thought here. Are you doing any of this stuff with your mice? Bill, are you doing any of this stuff with your mice? Are you doing any of this stuff with your mice?
It sounds more like me.
The way to get it, though.
You know how to use
Just take a gene in for whatever you want. I mean, that seems like that'd be it. Just take out and even knockout mouse instead of breeding for 10 generations.
Put
you
all
just
the different
CRISPR
combos.
out, yeah. CRISPR out the gene you need to take out.
That's right.
This is a scary place we're going here. Scary.
Yeah, this is trying to extrapolate 50 years from now. That's what would be going on.
Well, yeah, I mean, it wasn't very long ago that you could put any DNA in any human cell or any systems. Now, we're not only putting it in there, we're modifying it. Obviously, the field has changed.
Yeah, and then be able to do multiple knockouts, because not everything is mono gene kind of a problem.
I mean, you look at what they're doing now with Washington, where they're shooting it like this deficiencies. Free birds, you can collect them now in utero. It's like, and this is getting a risk.
Well, not all the cancers, too, that are all related to a certain
Did you read the article in the paper this weekend? Unfortunately, most cancers are random mutations. Like 99% of cancers are just random mutations.
Right, but some of them, like lung cancer,
Well, it fixed everybody now.
Yes, indeed.
they still have maybe 500.
Yeah, but it's just sort of depressing. I think it's just random mutations, which is anything about it. Did you put
So Ryan's been hanging out with Mason. I don't know
So
what
this cheese monkey mice that Bill has, they gotta be careful. It'll be a random mutation. I just read that going, God, this just sent me back 20 years.
They still work on the metabolic diseases first.
It's interesting that, you know, science doesn't go in a straight line. There's these big circles and offshoots of the line. Like this CRISPR stuff's going to get somebody to Nobel Prize.
I think there was a between MIT, Harvard,
It's supposed to be adjustable, so it's very contract. So Harvard has equivalent of the others.
Because they have more money. Got better attorney.
state cut.
I don't know.
So,
Yeah, it was good. Type of thing. But all garbage.
hours, they say you want to come to
Yeah, so we're discussing today. Wyoming. Yeah.
Oh, you went? You actually went? Okay.
We actually went. So this is a little video of their performance.
This
is their lead singer.
Okay.
Like, I thought this kind of music is dying.
That's what I'm saying. Is that when you